Everybody, everybody's back from the break, and appreciate you bearing with us despite our technical difficulties and having to shuffle things around. So I'm happy to introduce our speaker now, who's originally scheduled for 10, and he's happy to do it now. So Dr. Jake Chen, he's the Chief Bioinformatics Officer at UAB and Associate Director at the Informatics Institute. He's also a professor of genetics, computer science, and biomedical engineering. He did his PhD at University of Minnesota, Twin Cities in computer and information sciences. And he has an interest in biological data mining and systems biology and personalized medicine. He's also was elected to the American College of Medical Informatics. The name of his talk is AI in Cancer Research and Oncology Applications. Overview. Okay, so take it away.
Well, thank you. I'm really happy to be here and talk about this topic. And in a sense, this is one of my favorite topics to discuss because my lab setup here also has AI as one of the word letters in it. I want to actually just make a disclaimer today. I want to just talk about the brief overview, and I'm not going to talk anything related to my own personal research. However, I will leave some questions at the end to make you think, wow, how powerful this AI is. At the same time, there are lots of unfinished questions and challenges. So this way, you may actually come to my research talk, which is scheduled on August 16th at the genetic department at noon on August 16th, which is also Friday. So with that, here's my outline, and I'm just going through this. So I want to start with AI, and it's such a fervor in the past few years. Prior to 2016, before deep learning became very popular, if you do Google searches, it's really steady. But after deep learning become popular, and especially Google has this AlphaGo that to be the best Go player in the world, and people's interest in this technology has really doubled essentially through the internet searches. And if you go to a more specialized journal database like PubMed, you look at the publication that include AI, machine learning, and cancer. In 2016, it's only about 1,000 papers. Last year, it quadrupled. So researchers seem to be more interested in this application in cancer and other diseases. And that partially inspired us to do this data science hackathon that Amy is running wonderfully. And I hope that you also benefited from this. I wanted to do this talk a month ago, but because of other engagement, that I thought that maybe now is a better timing, because now you have all the other skills. You can reset your mind and ask the question, what should I do? And in order to really make a huge impact with this technology. And all of you probably know some real world application of AI. If you go back 20 years ago, this would be like a science fiction. You are sitting in the Tesla, and the Tesla has this computer already have a pre-trained model, and it used the sensor and the camera to detect all the scenes on the street and build some kind of a model of what are patients, what are cars, what are objects, what are clear roads, and do route planning as a result of that sensing in a real-time fashion, so that it also controls the cars, which is also a separate part of the engine. So that's incredibly smart. And I would predict that in 10 years, we don't even want to go back to the older technology. Who would want a car that is not smart, that is error prone, that is also can introduce systematic error. We would all like not only smart cars, but smart traffic controller that can talk to our car, and smart car-to-car communication so that we never get stuck in the traffic or have any traffic accident anymore. Right. So what exactly is AI? I would like to start with this audio, this video. This guy from Google, he can tell you a little bit more. And for maybe a little or 5 minutes, and see if the audio would load in the library network, which is not very promising now. Let's see. It does have the network. Can you hear the sound from the video? Oh.
I'll help you share. I can help you.
Like it's going to slow down anytime soon. Machine learning brings the promise of deriving meaning from all of that data. Arthur C. Clarke famously once said, "Any sufficiently advanced technology is indistinguishable from magic." I've found machine learning not to be magic, but rather tools and technology that you can utilize to answer questions with your data. This is Cloud AI Adventures. My name is Yufeng Guo, and in each episode we will be exploring the art, science, and tools of machine learning. Along the way, we'll see just how easy it is to create amazing experiences and yield valuable insights. The value of machine learning is only just beginning to show itself. There's a lot of data in the world today, generated not only by people, but also by computers, phones, and other devices. This will only continue to grow in the years to come. Traditionally, humans have analyzed data and adapted systems to the changes in data patterns. However, as the volume of data surpasses the ability for humans to make sense of it and manually write those rules, we will turn increasingly to automated systems that can learn from the data, and importantly, the changes in data, to adapt to a shifting landscape. We see machine learning all around us in the products we use today. However, it isn't always apparent that machine learning is behind it all. While things like tagging objects and people inside of photos are clearly machine learning at play, it may not be immediately apparent that recommending the next video to watch is also powered by machine learning. Of course, perhaps the biggest example of all is Google Search. Every time you use Google Search, you're using a system that has many machine learning systems at its core, from understanding the text of your query to adjusting the results based on your personal interests, such as knowing which results to show you first when searching for Java, depending on whether you're a coffee expert or a developer. Perhaps you're both. Today, machine learning's immediate applications are already quite wide-ranging, including image recognition, fraud detection, and recommendation systems, as well as text and speech systems too. These powerful capabilities can be applied to a wide range of fields, from diabetic retinopathy and skin cancer detection to retail, and of course, transportation in the form of self-parking and self-driving vehicles. It wasn't that long ago that when a company or product had machine learning in its offerings, it was considered novel. Now, every company is pivoting to use machine learning in their products in some way. It's rapidly becoming, well, an expected feature. Just as we expect companies to have a website that works on your mobile device or perhaps an app, the day will soon come when it will be expected that our technology will be personalized, insightful, and self-correcting. As we use machine learning to make human tasks better, faster, and easier than before, we can also look further into the future when machine learning can help us do tasks that we never could have achieved on our own. Thankfully, it's not hard to take advantage of machine learning today. The tooling has gotten quite good, All you need is data, developers, and a willingness to take the plunge. For our purposes, I've shortened the definition of machine learning down to just 5 words. Using data to answer questions. While I wouldn't use such a short answer for an essay prompt on an exam, it serves a useful purpose for us here. In particular, we can split the definition into 2 parts, using data and answer questions. These 2 pieces broadly outline the 2 sides of machine learning, both of them equally important. Using data is what we refer to as training, while answering questions is referred to as making predictions or inference. Now, let's drill into those 2 sides briefly for a little bit. Training refers to using our data to inform the creation and fine-tuning of a predictive model. This predictive model can then be used to serve up predictions on previously unseen data and answer those questions. As more data is gathered, the model can be improved over time and new predictive models deployed. As you may have noticed, the key component of this entire process is data. Everything hinges on data. Data is the key to unlocking machine learning, just as much as machine learning is the key to unlocking that hidden insight in data. This was just a high-level overview of machine learning, why it's useful.
Okay, well, I hope you get the gist of it, and it's almost perfectly built for cancer applications. And I certainly find when I browse this video, let me see. I certainly find it was a pretty joyful. So coming back to artificial intelligence, and you may actually be overwhelmed with these different terms. Since you're not a computer scientist who studied the history of computing, I'm not going to bother down with the details of the differences. But you can roughly say that artificial intelligence is a term that came up in the 1950s. And back then, the logic programming was, and reasoning capability was pretty much the majority form of intelligence. Because the computer back then didn't have a lot of processing power and we didn't have a lot of data collected. And then it came to machine learning that's pretty much built on statistics. And people are pretty smart in building statistical models to figure out how to do things without the reliance on huge computer power. Then it comes to deep learning. And the deep learning is something that we're gonna just talk next. So a brief review on classical machine learning models. You have some raw data, for example, DNA sequences, different sequences from different species. You can clean them, use the FAST presentation, and then align them together. You can extract features, for example, the DNA binding motifs, or just the domains based on homologies modeling and which position have which residue preserved. And then once you have those information, if you know that the certain patterns are used in a certain biological context, you can train a statistical model. Sometimes it's linear, sometimes it's non-linear, to actually learn how to classify the samples based on the labels that you train it with. And then you come up with the result. Right? So this is a very classical and traditional. In deep learning, however, it does things slightly differently. Instead of trying to force the users to come up with a list of features, in deep learning, you are actually asking the computer to do the bulk of the work. Right? So the computer come up with layers of a neural network, and then the neural network were employed to assign different weights and activating what they call the activating different nodes, just like the human brain, how the human brain theoretically works. And through these layers of convolution and subsampling, you can actually identify any possible permutation of features or feature combination that leads to a certain outcome. And the deep learning use supercomputer capabilities, and they're very efficient in just making sure that the weight matrices in the network can actually bring forth the answer that's exactly what you're looking for. So that's a feature engineering plus classification that sits between the input and output. And I won't actually go through this other video, but essentially talk about the difference between deep learning and machine learning. The major difference is the computational overhead. In deep learning, you need to actually train on bigger data sets because the neural networks are pretty hungry for data. If you don't feed it with enough data, sometimes you have this sampling bias. But then it requires a lot of computing power. Luckily for this contest, we have UBright and we have the supercomputer. We have give you all the tools. So if you're interested, I really encourage you to do some deep learning as opposed to standard machine learning, which probably perfectly okay if your data set is small. Okay, so with that, let's actually move to cancer research. And what are some of the big data opportunities? So in cancer, one publication that I really like is actually organized by this cancer journal of clinical oncology. And people are saying, well, we have big data in cancer field for sure. And these big data may come from next generation sequencing of patient samples or other omics such as proteomics, microbiome. We may have an electronic health record. We have lots of cancer related pathology images. And then we have cancer clinical trial information and even the basic science, how tumor works, where gene works and protein works. These are big data. And the outcome is very clear. People want drugs that works, individual progress, and the disease-free cancer survival. We want early diagnostic tests. We want to be able to sort out the patients so that they can receive individual treatment more accurately. So all of that relies on the mathematical modeling. And the AI is a very major component. What I'm not going to talk about today is systems biology, which is a different dimension, and I'll defer that to my August 16th talk. So one of the major data sources that we're not bringing into UBright, but hopefully many of you know already, is called the Data Commons at cancer.gov. In Data Commons, they have data collected through almost 100,000 participants over more than 100 large-scale cancer studies that involve close to 1.5 petabytes of data. So it's a very sizable data, even if your computer usually that just have 1 or 2 terabytes of data, you need 1,000 computers to be able to just hold the data. So obviously that's done in the cloud, and National Cancer Institute provide all kinds of tools. If you want to actually do this contest, you can register an account and use it over there. Right? And I think the NCI has this concept called the Nci Data Commons. And I really would like to bring to your attention. I think it's also a very neat audio that is worth playing. So let's see if it's playing without this.
When people think of the extraordinary breakthroughs that are made in cancer research, they don't realize the harmony that takes place behind that research. In music, this only happens when notes work together, build off each other, and combine to create the perfect harmony. In cancer research, data pieced together in a specific way can lead to new discoveries and treatments for cancer. Now, thanks to the NCI Cancer Research Data Commons, or CRDC, sharing data is becoming more harmonious than ever before. CRDC is hitting the right notes, providing access to many types of data. genomic, proteomic, imaging, clinical trial, and more, all in one place. It is enhancing the way data are shared through a single cloud-based and expandable infrastructure, one that empowers researchers to perform at peak by working in real time to explore and analyze data without the downtime of downloading, and equips them with the computational power that only comes with the cloud. CRDC increases connectivity, allowing researchers access to standardized and harmonized data, to ask more complex research questions by combining diverse data types across the CRDC or with their own for integrative analysis to increase our knowledge about cancer, all further supporting the goals of precision medicine, all furthering the effort to accelerate biomedical discoveries without missing a beat. The CRDC gives researchers a place where they can work in concert, in sync, and in harmony. Now that's music to many people's ears. National Cancer Institute, cancer.gov, 1-800-4.
Okay, yeah. So that's pretty impressive, and it's built with many informaticians and IT professionals' hard sweat effort. And if you actually dig out the Cancer Research Data Commons with a little bit more details, you see that it consists of 6 different repositories. Each repository have specific types of data. And they have the infrastructure, like the tools that help you analyze the data. And some of these tools may require some training before you can use it. But they also have the cloud resources provided through Broad Institute, Institute of Systems Biology, and Seven Bridges. And I believe that earlier this year, we have the ATIS meeting, and Seven Bridges have done some of their tutorial. Okay. If you zoom into the Genomic Data Commons, you see that they have all kinds of data that is very specialized. Some of them are microRNA, some are methylation, genotyping, RNA-seq. So many of you may be confused. So what kind of a data should I use for this data science hackathon? Now you know that even though we provide the convenience of downloading and analyzing your data in UBright, as Jendo wonderfully presented earlier, and you really don't have to use the data in the UBright. You can directly use the data in the cloud and maybe get a free cloud account, Amazon or Google account, and start working. Right? And then if you like a more traditional style website that downloads the data, you can go to CBio portal. That's on the left side. If you're interested in a specific cohort, like a children's cancer, then you can go to a data that's also captured by the CBio portal. That's not the TCGA, called the target. Or if you're interested in the international consortium of cancer data, there's the ICGC data portal that you can look up online. So there are many sources of the data. Specifically in cancer, TCGA is very popular and widely used. Here is a figure that shows the percentage of data available across different types of cancer provided by TCGA. So you can see the green ones are mutation, and then the blue ones are clinical data, and then there are copy number variation data, red and orange. So they almost have, all of them have lots of the omics data. The images data, surprisingly, are not that abundant for TCGA. In terms of the cancer studies that you can do, there's an online peer-reviewed paper published this year that talk about the popular AI method that people already apply to cancer. So as you can see, the ranked as #1, most people are using artificial neural network. If you're thinking about what techniques to use. And some of them use a clock regression model that's more traditional. And then some of them use other models and Bayesian models, more traditional machine learning. So you can see that you don't have to take deep learning as your objective in order to score. And most other people are not. But interestingly, when you look at the data set size, and there are different size associated with different cancer. The one with the largest size is breast cancer. The one that doesn't have a lot of data are just prostate cancer and these smaller cancer types. When you're looking at people doing either unique cancer-specific studies or pan-cancer studies, as you can see, based on by the publication count, most studies are actually focusing on a specific cancer. So I would just expect that if you want to take a less of a challenge for this contest, perhaps you can just focus on one cancer. Although there are singleton papers that are actually trying to study the pan-cancer gene signatures or biological mechanism that's uniquely available. And certainly that's encouraged, but I think that's probably going to be the exception. And there are also other databases that's more specialized. And for example, I look up this database called Nci. It's for single cell state atlas. And you see that these flowers actually represent clusters of cells that are grouped together. And they only have about 25 cancer types and 50,000 single cells. And so those kind of data sets are going to become flooding our field in the next few years. And they're just beginning to start. But those of you who are interested in these new novel sequencing types, I believe that this is also another good starting point. Obviously, we have this website, cancerubright.org. We're trying to put more information about the cancer data set. I'm pretty sure that Amy has already mentioned to everyone, please submit your data set resources. If you felt that this is something that we need to cache locally in Ubright to make free computing associated cancer analysis easier and faster. And we're happy to do that as part of the Informatics Institute. So that's the commercial. Okay. Now, if you remember at the very beginning, the Google lecture videos that I cited, the AI is really about the data plus the problem. So let's talk about what are some of the one challenges that you need to think about in cancer. One of them is of course, the individualized treatment of cancer patients, the so-called precision medicine. Suppose you have 3 patients in here, and the patient A have some mutations and that needs to use a drug A. You treat it, kill the cancer. Patient B on the same organ but have different mutations, you use a different drug, drug B. That's fair enough, but patient C have a different type of cancer, however, share the same mutation with patient A. Maybe you can repurpose the drug A for this, so that there's a drug and the individual patient, if you compare them, wouldn't that be wonderful? However, a few years ago, there's a paper that published that said, at least a few years ago, when you're trying to pair it just based on the target sequence mutation, matching tumor with drug target mutation has not been a very high yield activity except for a few cancers, like acute myeloid leukemia, where you have a very well-known mutation and drug target. But otherwise, and there they're all over the places, and we don't know which one is a driving mutation. So that outlined the challenges in the individualized medicine treatment for oncology. One of that is that the cancer is not homogeneous, even within the same patient. There's a paper published a few years ago, and they were taking samples from 3 patients, colorectal cancer patients, and they try to cluster the clones derived from different sites of the 3 different patients. And you can see, even though they roughly cluster, but when you try to take the different clones from each patient, try to subject them to the drug, and there are different colors representing different response to drug or the drug sensitivity problem, you will see that it's all over the places. Some drug for the patient, it work very well. Some drug actually depends on which clones you're selecting. And it's quite a mess, and outline that you really need to know a lot more details about clonal evolution of cancer in order to introduce effective treatment. So that's an unsolved grand challenge. Another grand challenge that's actually being addressed is to do cancer screening with cheaper technologies. Imagine if you have a relative, and you may have heard of them, they go to the hospital today, and the genomic testing is still rare. The more common testing is actually doing imaging. But a lot of the time, the imaging cannot be used for diagnostic purpose in the clinic. You take the images and you suspect it's tumor, and then you have to do a biopsy. So in this work, they actually try to say, okay, if I have images between the baseline and the follow-up, can I just look at the baseline and the image features, maybe the tumor microenvironment, there will be subtle features that even human may not be able to tell. Can you predict the 5 years whether this patient will have cancer or not? And amazingly, this AI does very well if you look at the ROC curve, and likewise for other applications. And so this is a grand challenge, and I think that we also have some imaging data in our UBright that the Jendo had provided, and you can play with it. So back to the question about the tumor evolution, clonal evolution. And it's very complex. If you go from the top to bottom, right, you start from the top, the tumor was just not even a tumor, it was just a mutation introduced. And the cancer will actually multiplicate, the tumor cell will multiplicate into different clones. And then there's a lot of heterogeneity. And as you have more and more, you have subclone that can outcompete others. And then their huge expansion get detected by the immune system. And then they try to kill it. And then the tumor also try to fight back. In that process, if you introduce some treatment, a minor population of the tumor would survive and continue to expand into new clones. So this is very tricky, right? So we can't even find the enemies in the battlefield. How difficult it is to track down Al-Qaeda leaders in the battlefield. And that's $1 billion or $1 trillion effort. And imagine that we're doing this on a small scale for all different kinds of cancer in the patient. So we're pretty much shooting in the dark. Another thing is that trying to use cancer immune defense mechanism to develop newer therapeutic solutions. So for example, whenever a cancer evolves, it come up with all different weird proteins. And as cancer are just attacked and digested by different immune cells as the first line of innate defense, and those immune cells actually will present so-called antigens, and that's not actually in the body itself, and will be recognized by T cells, and the T cell will expand, and actually then and that the clonal expansion will actually trigger other immune system to actually pay attention to the situation. But if you can actually predict those new antigens, and you may actually be able to proactively develop vaccines that will present to the immune cell surface and activate the T cells. And even if you have other cancer in the early traces, and then you'll be able to wipe them out. So that's a really nice thing to have. And there's a lot of a computational challenge. How do you sift out all the noises and just look for interesting signals and the real signals? Now, another challenge is to integrate the multi-modality of features. As I mentioned at the very beginning, TCGA dataset, even that dataset has multiple data types. You have a copy number information, you have EHR, you have imaging, and sometimes because of the weakness, data coverage or noise, or the individual aspects of the tumor, you don't get a whole picture just by using one type of a modality. So you need to actually combine multiple modality together. And so this review paper pretty much shows that if you have 3 types of data and the protein expression, genes with somatic mutation, and some copy number variation in the pathway, you can actually try to build multi-modality models and use this line graph to indicate the association relationship between features, and then use that association as additional features for machine learning tasks. And the bigger challenge is actually outside of the cancer research, but in the clinic. As the tumor evolves from the initial onset, tumor evolution, and maybe resistance subclones, you have lots of subclones. And at the same time, the doctors are trying to intervene and try to treat it. So there's a question is, can you detect the cancer early before they're metastasized? Can you diagnose the cancer with cheaper technologies or even blood tests or imaging tests? Can you come up with therapies like immune therapies, better immune therapies? Can you individualize it? So these are all the grand challenges. And I would like to point it out to you so that you don't go to the dark alley when you're doing this hackathon. You may not get there because it's only 40 hours, but I hope that you understand the judges sitting behind the closed doors, they understand cancer. They're gonna judge your computation based on the grand challenges, but not on how fancy you were able to come up with the machine learning techniques. Okay, let's look at some of the success stories. So in detection, for example, you can say in the lung, in the breast, in different parts of the organs, you can detect the suspicious regions, and usually they're false positive. They're benign for the most part, and then you actually separate the true positive, the real cancerous ones, from the false positive ones. And the second is how do you characterize those cancer samples and images? You need to do some segmentation, separate the tumor from the background context. You need to stage the cancer depending on different stages and then have different assumptions and so on and so forth. And the more, a lot harder to do is to capture the dynamics. If you have the same tumor, if you take images twice in different time point, can you keep track of the changes and monitor the changes, right? And how are we performing against the actual pathologists? Actually, a lot of the research, if you do it well, you can do better. So this is the statistical performance comparison chart of a task try to predict 5-year survival from pathology images for colorectal cancer patients. There are 400 patients involved, and you use a deep learning method versus the visual histological assessment methods done by the pathologist. The deep learning methods are the yellow ones, okay? And the pathologist methods are the regular yellow. No, the orange ones is the deep learning, and the yellow and the blue ones are the pathologists. So in every statistical measures used here, deep learning seems to be performing a lot better. And there's another one done by another group that try to look at myeloma detection in skin, right? So they use the best convolutional neural network deep learning method and achieve 86% of accuracy. That's the machine performance. And for the same job, they asked a pool of beginner pathologists, and the beginner pathologist was maybe just like me and was 30% accurate. Skill ones, you add another 19%. Expert one, you add another 51%, but nonetheless, it's only 82%. So machine versus man, it seems that AI wins in this contest. And then, of course, you can build multi-modals, and this is another research that talk about building multi-modals to take account of electronic medical record data, copy number variation, and gene expression profile together. And this is the red performance using ROC curve. It's a lot better than if you were using them separately. Okay, so that's really encouraging. And increasingly, there's more research being done on multi-model analysis. If you see the paper where you have 2 dots, that means you are using at least 2 data types. And there are many papers that are publishing using gene expression, which is a majority. That's not surprising. Gene expression, microRNA, gene expression, and other types. So I highly encourage you, if you want to win the contest, and maybe consider more than one data type to overcome the weakness. In the end, I think the promise of clinical application of cancer is that you can actually do AI enhance the clinical workflow. And I don't think that there's anyone who think that the AI is ready to take over the pathologist or the doctor yet. Maybe it will happen in 100 years or 50 years, I don't know. But AI will certainly help improve the process. The traditional one is listed on the left side. So you have radiology assessment, and the doctor will actually look at the clinical factors, patient preference, come up with a treatment plan, do some biopsy, do some gene expression or gene analysis, and then you determine the clinical outcome with time. And that is very, very long time. For AI-enabled new treatment paradigm, AI is going to come in at every stage to help inform the physicians. Sometimes they're overwhelmed by the information, to help them make better decisions. So that's the whole idea. It's never meant to replace the patients or replace the physicians for all the literature that I have found. Now, I want to quickly just say that we have done a grand challenge contest to the community that may be inspirational to this data science hackathon. It's not done in the cancer context, but everything else is very similar. So we call it the rheumatoid arthritis Dream Challenge in collaboration with pharmaceutical company, Sage Networks, Mount Sinai, and other universities. And the contests were very easy to define. The problem was that in rheumatoid arthritis patient, their bone structure, especially hand and feet, will deform over time. And they will deform in different shape and forms. But the two major ones are joint space narrowing. So the space is so tight, they can't move their fingers. Or there's a joint erosion that essentially will become so deformed. And that's one of the main disabling effects of rheumatoid arthritis. So the grand challenge is, can you actually just read the images and then score it automatically based on machine learning? And we thought previously, getting labels, whether to tell this is disease or not disease, that should be very straightforward. But how do you score it? So we decided to put up a grand challenge, and we collected this data, and this data was actually collected through Lou Bridges, one of our collaborators before he moved to New York City. I'm not sure that's a good idea given the pandemic. But he moved there. Before he moved there, he has lots of cohort that provided the basis for this interesting AI competition. And these are the images, and you can actually, you know that there are a lot of work done in curating these images with joint narrowing score and erosion score that you can actually use to inform the machine learning algorithm what kind of the training that can be done on existing label data before you apply to new data. So we set up the challenge, and interestingly, we have contestants all over the world and multiple teams. In the end, they go through some leaderboard. There are more than 100 teams, and then, no, I mean, not more than 100 teams, more than 50 teams, and then we trick it down to less than 15 teams that we invited for the final round because their performance are good. And all the computation were done on the UBI platform, UAB supercomputing. So we can do that, and I'm pretty sure that you can do it for your data science hackathon. And then this is a baseline, and then the winners are just picked. We're not doing that strict, so there's no objective judging. And the fact that everyone will be evaluated differently in this contest, but you get the idea. So we have some lessons, and when we look at the winners, I thought that those lessons are interesting for me to share with the audience here. You got to actually perform data pre-processing. The data are not AI ready to begin with. If you think that is a data that maybe Matt or Jendo is provided to you is a holy grail, is gold standard, think again. You need to actually, even if you use images, you need to standardize the images so that they're the same quality. You need to do the so-called data augmentation because the machine learning, deep learning require lots and lots of data. You usually present it just with a few hundred data sets. You need to use a proven method to just combinatorially generate similar data to feed the computer to train. And then you need to do image segmentation. In genomics, you have to process them using gene expression analysis pipelines or mutation calling pipelines. And that's all the bioinformatics knowledge you have in your team will be essential. Don't forget that as you're jumping into the AI bandwagon. And the second is that you really need the AI expert. The data mining model architecture choices has a lot of impact on the outcome. We've seen that in the context, a lot of teams fail, not because they use deep learning, but because they use the wrong architecture. Parameter tweaking is also required. It's not like a magic box. Usually, if you want to try the best one, you probably need to come up with independent solution and try to use ensemble method in order to get a better performance. Last but not the least, I want to talk about the hypes and the traps. You have a computer, and computer can make stupid decisions faster than you do. Yeah, they can be smart at times. AlphaFold 2 is one of the poster child of what AI is able to accomplish today. If you didn't know about it, you recall that in a week or two, I mentioned in one of the Friday seminar that if someone look at what Google DeepMind have come up with, and you'll be impressed because in AlphaFold 2 is able to almost predict the protein structures and look at the protein structure solved by some of the best labs in the world. There's a Young John's lab from Michigan. They went through the competitions, and then the real structure versus predicted structure, they don't align very well. David Baker, very respected researcher from University of Washington, doesn't solve this problem very well. AlphaFold 2 somehow have amazing alliance of the truths and their prediction, to the point that even nature said it changes everything. It solved all the protein structures, and last week, I think they make an announcement that 300 protein structures predicted through AlphaFold 2 has already been deposited into the UniProt database. It seems that the problem has solved. You go there, you can find it. Until last week, actually yesterday, in my group meeting, by the way, actually that's a journal club, and everyone are welcome to come to the journal club next in the fall. We'll have lots of fun reading those papers. So we actually picked this database, and we come up with a protein structure called WASP. You can just explore it yourself. And the WASP has 3 domains, and the Google AlphaFold 2 predicted 3 domains very well, but the rest of them are pretty much random. And they're good regions, and the white regions are pretty much not predicted at all. So you can see that there's a press is one thing, but when you go down to the reality, there could be a lot of variability in the solution. Do not just trust AI blindly. And I think, yeah, we learned that every technology, there's expectations and a hype. Also, there's a deeper issues, and I'm showing you 4 images. Can you tell which images is fake? You're not a pathologist, you probably don't know. In fact, there's only one real image that's actually A. And this B one is a synthetic images generated by conditional generative adversarial network, or the GAN infrastructure. It's almost like the real one. And then this C is a randomly drawn image generated by computer. And then the computer can generate synthetic image from this randomly generated one that looks alike a regular pathology image. So imagine that you're studying this, you go pull out some internet website, and people don't tell you where it's coming from. It's not from credible source. You have to think twice before you apply it. You may contaminate our knowledge. You may actually, if you get approved without the expert, you may actually, instead of saving patient life and hurting people, right? So those are the things you want to avoid. So that a lot of the weakness of the technology brought the trustworthy issue. There's unintended consequences of using machine learning blindly in medicine. There's a geographic biases. That's why NIH just came out with an Aim Ahead program that try to say, well, wait a second. We want the people in Alabama also provide the samples and contribute. We cannot just have people from California and East get their samples and then claim that the problem is solved. In fact, that's reflected in our NIH grant proposal. We had an amazing team from top medical institutions got rejected, but our Alabama team were invited for proposal writing. It's really interesting that NIH is well aware of these problems. And there's also algorithm selection biases. That's why you need to use ensemble models, right? So improper use of the AI in the clinic, as we said earlier, could reduce the skill of physician physician could become lazy, thought all the problems were solved, and they may focus just on the few texts that the AI generated for us and forgot the context. And also the worst thing is that you don't think about the unknown unknowns, which is pretty much the art aspect of a medicine. There's a science aspect that we're trying to do, and there's the art aspect, the caring aspect, and finding out what is not captured in the data that make it more impelling, imminent, that we don't treat AI as a black box, but instead we use AI to interact with humans and help humans make better decisions. So this is going to be my last slide. So I think that AI is very promising. It can handle many complex nonlinear relationship problems in cancer. There are many exciting applications. I hope that you can generate some really good ideas in the data science contest. But there are issues, and it's not a magic bullet. So I hope that you don't overclaim your contribution. And this talk is not possible without me talking constantly to many biologists and my people in my group that constantly bounce the ideas. Going 5 years ago, and then I probably know very little about the technology application and precision medicine. But I think in the team, I'm able to learn a lot faster. So hopefully you get the message. We want you to actually write down the teams, build teams, very diverse teams, and then that probably would be the essential for you to win some challenge award money. And with that, and I apologize a few minutes of overtime, and my daughter is playing nearby. I hope that you get some information out of it. Thank you.
Yeah, thanks for that very enlightening talk. It is encouraging that we can maybe have more light at the end of the tunnel than that paper that you cited that it was a dead end. Anybody have any questions? And either about the hackathon or about this talk?
I have a question. So it looks like there's a lot of work involved in getting this project to be polished, but we only have 2 days. So how do you have like the real expectations for that? Like, would we just have like the idea and show some promising results, or do you expect like a paper ready, publishable results, or so? What are your thoughts on that?
Yeah, that's a very good question. I think that everyone, there's a Chinese saying, 八仙过海, 各显其能. There's 8 gods that's trying to cross the sea. They all have different skills, and then they find their ways, either flying or zooming through some stick, magic wands, or sailboat. So you should look at your expertise and who you can assemble in the team and the mentors, and assess what is the best strategy to get the best outcome. Some team, like some of my members of the team, because we're doing this work, we have some prior result. Of course, if we're going to use the new data, we will have certain advantages. But other team, because we stack this with a lot of people from the traditional knowledge discovery KDD conference, they may be invisible. You really need to look at the registration expert table and target them out or yell out in the Slack channel. I need this, and I have a great idea. I'm looking for a solution. Or another person saying, I'm looking for great cancer challenges, and I have great skills. Find your way out. We're here to help you, but then we know how to do it ourselves, but then you have to actually help yourself by working tirelessly before this 40 hours of the 2-day hackathon. And the key is the preparation, and the 2-day is probably, once you say ready, it's like a chef in the kitchen. You need to prepare the preparation may take you days, even starting from growing the right cow, selecting the cow, and then make them into the beef. And then you can probably serve the best gourmet steak. Okay, I hope that I'm answering your question.
Let me rephrase my question. So would like wonderful ideas, but not so polished AI kind of method would be okay? Or would you rather have like very like conventional ideas, like well-charted areas, but like kind of like not that innovative, I would say, but
I think
like very
everyone,
established
I
pipeline.
got your idea. So I think it sounds like that we have two purposes for this data science hackathon. One is everyone is together, we're here to learn and have fun, and you're not here for the contest. For that purpose, I think any idea is good, even out of the box ideas. Just have fun for that 40 hours. It's like a marathon. Most people are not in the marathon for prizes, but for the few groups who are after the prizes, and I think you may be up against very intense competitions, and you probably need to have the best data, clean the data, and a great idea in order to win. I don't know the whole quality. Maybe in the end we're so disappointed that there's no ideas, and we need to go through the whole 2 or 3 years of training as a bioinformatician. That may be okay. I don't really know what is going to be the outcome for the best team. We just know that you're all encouraged to just learn and have fun in the hackathon.
Yeah, I want to emphasize that we know it's 2 days. It's to come up with and develop an idea and some way to solve a problem. We definitely don't expect fully finished ideas. We don't expect anything that's publication ready. And you know, you will present to us what you're starting with, and then what you've accomplished in 2 days. And so the judges are not looking for, oh wow, this is the most flashing, shiny object kind of thing, or the most elaborate thing. They're looking to see what kind of progress that you've made. Just like if you're learning music, you would the progress you make would be important. So if you're a professional musician, and if you're an amateur musician and the professional someone who would learn something and didn't really change that much, you'd be like, okay, well they're really good and polished, but maybe they didn't progress that much. But if you're an amateur and you made a huge amount of progress, then that's very significant. That shows progress. That shows hard work. So sorry, I think my analogies are not as good as Dr. Chen's analogies. Any final questions? Because I know. Well, actually, and I think Zandos has stuck around too. So if you have specific questions about that, then any questions about that? All right. Well, I'll conclude with a few final announcements. I know I probably sound like a broken record, but if you're participating in the hackathon, then we really want you to really be proactive and get ready and enter your information in, join our Slack channel so you can discuss things, join the Airtable so we can see you. If you want to be eligible for competition, you need to have your information in Airtable. That helps us to get everything ready. That's the list we're using when we contact people about getting them access to things. And we want you to it's been kind of quiet there. I think everybody is kind of, I don't know, shy or busy or whatever. I know it's summer, probably people are on vacation and getting ready for the new year. But put yourself in there. There's the link in our Slack channel for how to get to the Airtable so that you can edit your information. And it's also on our website where you can submit your project, and you can also select team members. And Zandos mentioned earlier that if there's a public data set that you want to be available in UBright, then you put that data set in that list. I know it says prepare a data set. It's also prepare or suggest a data set. So we just want you to make sure to get ready. And our next Friday at 10 o'clock, we have our final networking and readiness session. So this will be the last opportunity to sort of make sure that you're all technically ready, and but you should be starting it now, and also and make sure that you're and try and work with your team, discuss your projects. This is the last opportunity to do that, and I really look forward to collaborating with you all and learning together. Any final thoughts and questions before we end this call? All right, hearing none, then thank you very much for participating, and we look forward to seeing you at the hackathon and at next week's event. Have a great weekend. Goodbye.
Thank you.